Hello, I'm Leigh Johnson from Brigham Young University in Provo, Utah, and I'm excited about the presentations that are going to occur today. And in the interest of time, I'm just going to jump right in. So most of us are familiar with the fact that California is the center of diversity for a number of genera, not just in Polemoniaceae. I'm going to focus today on the genus Navarretia, which has several instances of cryptic or near-cryptic diversity that have led to an underestimation of species diversity in this genus. So to begin with, there are several different patterns of crypsis, and I have to detail these quickly, but basically sibling or sister species might be what might be most expected or most common in terms of cryptic species. And the idea that we have one species that diverges into two separate species, and those two separate species are very similar morphologically. And it's easy to see how that could occur if the criteria that we use to recognize species evolve differently through time. And one of the last things that diverges is morphology, and it occurs much later than maybe even reproductive isolation. And of course, we would have morphologically very similar species. A good example of this is Paradoxonota and Paradoxoclara. Paradoxonota occurs on the west side of the Great Central Valley, and Paradoxoclara occurs on the east side of the Great Central Valley. Both of them are restricted to serpentine soils, and if you look at them, you can tell them apart based on their flower characteristics, different size flowers, different degree of stamen exertion. And so you say, well, are they really cryptic? Well, if they didn't have flowers, you wouldn't be able to tell them apart morphologically. But what really makes these species interesting in the situation or the case of cryptic species is the fact that they're closely related and morphologically resemble something that's not their sister species. In a case like this, we look at the things that are morphologically most similar and trace back their common ancestor, and we see that that same common ancestor gave rise to a lot of diversity that isn't morphologically similar at all. And this is the case with Navarretia Paradoxoclara and Paradoxonota. These two species are morphologically most similar to Navarretia intertexta, which has long been recognized. And actually all of the earlier collections of these two species were just identified as Navarretia intertexta. Navarretia intertexta can occur on serpentine, but it certainly doesn't have that affinity, and it occurs on many other soil types. And the common ancestor of Paradoxoclara and Paradoxonota with Navarretia intertexta actually also gave rise to the core vernal pool species, including Navarretia prostrata and Leucocephala, things that are morphologically really different. Another good example of some species that aren't siblings to each other, Navarretia sinistra and Linearifolia. If we look at the phylogeny, their common ancestor is actually the common ancestor of the rest of the entire genus of Navarretia. Yet morphologically, these two species are really difficult to tell apart. After a lot of looking over a lot of time, we came up with the fact that you can tell them apart by mature seed color. You can tell them apart with scanning electron microscopes of their pollen grains if you have one of those in your pocket. But with further looking, there are actually some other subtle differences in the habit, the branching structure, and the distribution of glands on their leaves. But this remains probably in my mind one of the most remarkable examples of cryptic speciation in the genus Navarretia. Another case that can give rise to cryptic species is when we have a clade or a group of species that have radiated relatively rapidly and perhaps recently relative to other things in the genus. And that rapid radiation has given rise to plants that have some reproductive isolation, either because of ecology or distribution or even intrinsic biological reproductive isolation, yet they're not morphologically very distinct. One example that fits this is Navarretia pubescens, and then the more recently recognized Navarretia gowenii and Navarretia pinocensis. The latter two species, in terms of on herbarium sheets, are represented rarely because they have very narrow distributions. And those that were first collected were identified as Navarretia pubescens because they fit that species more closely in the key than any other species. Yet you can see that we can differentiate them morphologically by stamen exertion, flower coloration, some other features with glandularity, But molecularly, these species are very, very difficult to tell apart, and there's hardly any differentiation, at least in the common genes. Certainly there will be molecular markers that will be able to distinguish these, and we're starting to explore some of those. Another case of cryptic speciation occurs with Allopolyploidy in some cases. If we have two diploid species that hybridize and form a tetraploid offspring, if those two diploid species are morphologically really different from each other, the offspring is usually intermediate and readily distinguished from both of the parents, which are easily separated. But if our parental species are actually morphologically similar to each other, there's not a lot of morphospace for that offspring to fall into. And in that case, we can have very difficult to determine species. A good example of this is Navarretia vividior. Navarretia vividior has until recently been considered a subspecies of Navarretia divaricata. And in exploring that to see, there's a question I had whether these should be recognized as separate species or if one really was better recognized as a subspecies of the other. I sampled Navarretia vividior across its geographic range, and in the process ended up identifying both of its parental species, Navarretia aeroides and Navarretia modacensis, which are recently described. And we can tell them apart, not just on chromosome number, but by morphology. Although Navarretia vividior is actually very similar morphology to its parental species, Navarretia modacensis, and those two can be difficult to distinguish. This ends up being important, though, if we look at the geographic ranges. Navarretia vividior as a subspecies of divaricata is represented by all of the gray marks on here. Once we have our species limits revised, we can see that modacensis occurs in a very different geographic range for most of the vividior, which is shown in purple. Aeroides is in blue. And then there's another entity here, which I haven't mentioned, Navarretia toriella, which actually isn't implicated in the origins of the vividior at all, but on herbarium sheets, that's where it has keyed out to. And so herbaria have a large representation of Navarretia toriella, which has been misidentified as Navarretia vividior. But the two are easy to tell apart, and certainly if they grew in sympatry very often, they would have probably more recently or been recognized as different more distantly in the past. Navarretia propinqua, long recognized as a subspecies of Navarretia intertexta, has been elevated to a species because we've shown that it's a tetraploid also. And it is not only related to Navarretia intertexta because Navarretia intertexta is usually the paternal parent of Navarretia propinqua, but it's equally related to Navarretia farnesii, its maternal parent. In the process, again, of looking at Navarretia divaricata and subspecies of the vividior, we sampled Navarretia divaricata across its geographic range and ended up identifying two additional morphologically differentiated entities that deserve species recognition, Navarretia crystallina and Navarretia miwakensis. These two species are of interest because they've specialized on pyroclastic soils and are restricted pretty much to that substrate. And then some other things that are kind of interesting. San Diego County has long had Navarretia peninsularis listed as occurring there. I've recently just finished reviewing all of the material that I can find that's been identified as Navarretia peninsularis from San Diego County, and Navarretia peninsularis does not occur there at all. Instead, all of the herbarium sheets represent either Navarretia modacensis, aeroides, or divaricata, and all three of those species occurring in San Diego County is actually a pretty good disjunct from their normal distributions further north. And there's still more work needed. Without going into the details, Navarretia attractyloides and other species have variation across their geographic range, which might represent new species. And then we have the Navarretia leucocephala complex and other species in the vernal pool clade that certainly need more work to help distinguish where the limit should be between subspecies and species, and can be difficult to identify because the features needed are the micromorphological features they possess. And I'm hoping that the species diversity that we've described isn't just the tip of the iceberg, that we're well on the way down on our iceberg in terms of understanding species diversity in Navarretia better. Thank you for your attention. Good day.
Hello, botanists and fellow plant enthusiasts. I'm Dr. Justen Whittall, and I'm excited to be recording this from my garage in the safety of the Santa Cruz Mountains in anticipation of a symposium on California, the importance of genetics in California plant restoration and reintroductions. Let me start by introducing myself and my lab by way of which plants we have studied in the past and how those fit into a world of adaptive radiations and cryptic species in particular. I come in from the Department of Biology at Santa Clara University, but my master's degree was in monkey flowers, Mimulus, Moschatus alliance, then my PhD in Aquilegia, the columbines. We've been working on Erysimum, Streptanthus glandulosus complex, some work on West Texas pondweeds, the San Benito evening primrose, and even in the Torrey pines of the Channel Islands and Southern California. But I think of these things, and I think in this conversation about cryptic diversity, we should think of them along two axes. One is the degree of molecular divergence or genomic divergence from low to high, and contrasted with the degree of morphological or ecological divergence from low to high. I can take these examples that I published on in our lab and from California and try to place them qualitatively on this axis, where over here in the upper left, we've got organisms or lineages with very low genomic divergence, like the columbines, yet exciting and interesting and important morphological variation and ecological adaptations. Same thing with the Erysimum, most likely in response to herbivores and soils. Streptanthus glandulosus complex is right around there, where there's very little molecular divergence, but a lot of exciting ecological variation. On the other extreme, we have lineages with very high molecular divergence, but very low morphological variation. The San Benito evening primrose is extremely difficult to differentiate from its closest relatives. West Texas pondweeds are another example of cryptic species, and the two lineages of Torrey pine, mainland versus island, another example. I suggest that maybe if you put this line, this one-to-one relationship line that the monkey flowers I worked on for my master's, the Mimulus Moschatus line, it worked out nicely and that the morphological and the molecular data seem to coincide. But I'll argue that in general, the pace of morphological, ecological, and genetic divergence rarely coincide with each other. In that case, we've actually got two main groupings. The grouping up here on the upper left, which I'll call recent adaptive radiations, and the group on the lower right, which will be our cryptic species. Now, recent adaptive radiations get a lot of attention. They're usually charismatic and macroflora, for example, whereas cryptic species are by definition underappreciated, if not downright unknown. So let's dive into them. Where do we find cryptic species? I thought that was a paradoxical place to start because by definition they are cryptic, but maybe we'll start in large families or large speciose genera like these top 5 in the California floristic province. And let's cross-reference that with lineages that span traditional biogeographic boundaries like the Central Valley or the Transverse Range. Now that said, that's where we'd like to start with our study of Caulanthus lasiophyllus. It's in the Brassicaceae and it's found across California. It actually goes to British Columbia, east to Colorado and New Mexico, and south to Baja and Sonora. However, this was an accident. Sharon Strauss from Davis and I were out collecting Jewelflowers late in the season. We accidentally picked up some Caulanthus lasiophyllus seeds from multiple populations, and we then started extracting DNA on the plants grown from there and found that there was a lot of genetic divergence between populations compared to the Jewelflowers that we were studying. We'd hoped it had to do with the fruit angle, pendant versus more erect. Turns out that's variable within populations and even within individuals, as you can see on the left, pendant versus erect fruits across developmental timings. So this is the types of questions that we can answer now that we're on pursuit of the morphological differences between genetic subdivision of C. lasiophyllus. We want to ask if it correlates with biogeography. Does it correlate with morphology? And does it correlate with climate? Luckily, I had some assistants, some undergraduate assistants. That's Tim Butler on the left, Stephanie Kim in the middle, and Jason Buenrostro on the right, all out in the field collecting Caulanthus lasiophyllus. We sampled 52 individuals from 15 field sites, and then we went to UC-JEPS, where they generously offered us specimens to both measure morphologically and sample for their DNA. We use the ITS, the TRN, and the NDHF regions. The last two are chloroplast. And we looked at 13 morphological traits. 9 of them were quantitative and 4 were qualitative. They were the most common traits used in keys to these lineages. We also included some close relatives, C. anceps, and C. flavescens, the closest relatives, which you'll see data on in a second. So the circles indicate a group of samples that have 8 ITS differences from the triangles, which are quite different. The circles were called the north coastal lineage, and they're typically concentrated in the north and on the coast, but they come down here to the on the coast to Southern California, Channel Islands. And the triangles are the southern inland lineage, which also was detected in parts of southern Nevada and Baja. The data that speaks to this is primarily the ITS data, and I'll highlight 4 columns here. Some of the columns represent multiple sites. We have 8 total sites that are 100% different between the coastal lineage and the inland lineage. Now, within each of these lineages, there were unique haplotypes, but they tend to be defined by a singleton, that's a single base pair difference, and not consistently differentiated or biogeographically so. A couple other interesting discoveries along the way are that the 8 differences in ITS between these lineages of lasiophyllus are 8 times more than the amount of variation we found between the 2 closest relatives, that's C. anceps and flavescens, 1 to the north, 1 to the south, and those are well identified morphologically, characterized, and no one's ever questioned their species status recently. We also found 2 samples, one from the Kesterson Wildlife Area in the Central Valley, Merced County, and the other one from the Carrizo Plain area of San Luis Obispo County that had a hybrid pattern. They had a variation in their ITS types. We looked at the 13 traits morphologically. We put them out into principal components space. And the first principal component explains about 1/3 of the data, but all of the characters are in the same direction, indicating this is a size axis. And so principal component 2 loads heavily, though, for fruit traits. And we did find a significant difference in the length of the fruits and in the pedicel in fruit. It's only about a 30% difference. And there's a big difference between a significant difference in a morphological trait and something that consistently identifies this, which that does not. As you can see from the PCA, these are wildly overlapping. Now, principal components axis 3 did not significantly differentiate these 2 lineages, so we didn't include that, but we looked at PC4 versus 2, and you'll see as we go down these axes, PC4 now loads heavily with leaf traits like size and pubescence. It explains about 1/10 of the data, and there's widespread overlap amongst these 2 lineages. So by definition, cryptic species using these traits. We did look at the climate to see if the climate was different for the 2. So we're now looking at monthly precipitation on the y-axis across the 12 months of the year using the PRISM data. And you'll see there is wetter, wet months in the northern coastal region versus much drier in the southern inland. If we sum that across the years with a new axis on the y over here, it's a 2.2 times higher precipitation annually. And that's wildly significant in a t-test. We grew these plants from many populations, and I have pictures from 2 of them in the southern inland and the 2 of them from the northern coastal. And we started identifying some really subtle but important morphological differences. For example, when the first leaves that come out, you'll see the lobing is quite different. The lobes are shallow in the southern inland region versus these deep lobes from the northern coastal region. Unfortunately, and we define this as the leaf sinus depth, which is indicated there. But it's only indicative on the basal leaves, not on the cauline leaves. And these basal leaves wither and dehisce during the transition to reproduction. So most herbarium specimens don't have this character, making it a little bit more challenging to extract from the specimen data. A couple of unanswered questions that I'll leave you with that we are pursuing are whether or not these 2 things are reproductively isolated. Although we tried to make crosses, the pollen had already dehisced in bud. So I point you to a great new study from Mimulus decorus by Coughlin et al. In Current Biology on a cryptic species with reproductive isolation. Are there cryptic physiological differences that might reflect these differences in climate? That would be fascinating. I was reviewing Clausen's Stages of Evolution of Plant Species who had the same thoughts. And how subdivided is the rest of the genome? We size the genome. It's only 3 times larger than Arabidopsis, and it's probably one of the smallest ever reported in the Brassicaceae, definitely in the Thelopodiae. Lots of people to thank for this. I want to point out Tim Butler, who is now Dr. Tim Butler, he's a postdoc at the Sanger Institute, Cindy, who's now Dr. Cindy Dick from UC Riverside, and Brody Sandell, one of our newest ecologists at Santa Clara University, who helped with the climate data. Khalik Dejan, Gavin, Julie, and Inez, who are really helpful in the field, and this has recently been accepted in American Journal of Botany, so you can read more about it there. Thanks so much.
Hello, everyone. My name is Kyle Christie. Thank you to CNPS for inviting me to speak today. So I'm currently a postdoc affiliated with Michigan State University and Northern Arizona University, but some of my work with Jewelflowers that I'd like to present today was done as part of my PhD at UC Davis with Sharon Strauss. So we're talking a lot about plants today and cryptic diversity, but I just wanted to start us off with an extreme example. So as recently as 20 years ago, we believed that, there was a single species of African giraffe. And so as we looked closer and learned more about the biology of these giraffes, it actually became apparent, well, maybe there were more than one species. So giraffes from different regions showed slightly different food preferences. They're occurring in slightly different habitats. They're mating at slightly different times, and they're mating with respect to the coat patterning. And so all of these things, all of these biological differences are actually manifesting in these genetic differences. So if you look on the left of the slide there, you can see distinct clades or distinct groups. So I thought, well, if there can be hidden diversity in these charismatic megafauna, there certainly can be hidden diversity in small plants. Next slide, please. So I'll be talking about the Jewelflowers, Streptanthus. So another example in mustards. So these plants are mostly restricted to California, and we say there's roughly 35 species. And as we look more, we're finding more and more species and hidden lineages, and we'll talk about that more today. But one of the really interesting things about this group is the types of habitats they occur in. So very rocky, barren, dry places. And the extreme example of that is serpentine. Next slide, please. So as many of you know, as plant enthusiasts, the California floristic province that I show on the left portion of the slide is a worldwide biodiversity hotspot. So extraordinary levels of species richness and also species endemism. And a lot of that really unique diversity is related to the presence of serpentine in California. So this unusual substrate, essentially uplifted oceanic crust. It's very harsh for plant growth. It has this very patchy distribution across the state. But while it only covers, maybe 1 to 1.5% of the area in the state, it hosts this disproportionate amount of the unusual plant diversity in California. Okay, next. So we look at this, this evolutionary tree or our hypothesis of the evolutionary relationships within the Jewelflowers. Serpentine endemism has evolved in a few places across the tree, but that group or that clade that I've highlighted in blue, every single species in that clade is entirely restricted to serpentine. So for today's talk, we're going to focus just on the single species, or as you'll see, species complex, Streptanthus breweri. Okay. So Streptanthus breweri, as it has been described, occurs over a really broad north to south range, maybe 500 kilometers. So that's unusual in Streptanthus. There's maybe only one other species, Streptanthus glandulosus, that has such a range. And what that really means, due to the moisture and temperature gradient in California, is that these plants or these geographic clusters are occurring in very different habitats. So in the north, in the blue populations, sort of the Shasta Trinity National Forest, it's wet, it's cool. We see these serpentine openings in conifer forest. As we move to the south into the Coast Range, in the turquoise, I don't have a picture, but those are essentially openings in the chaparral. It's a little warmer, it's a little drier. And then as we move further south, we're getting into these hotter, drier conditions. That picture on the left is some extreme serpentine badlands in the Clear Creek area. So we have a lot of environmental variation, and we've also noticed that, okay, there's also a lot of morphological variation in Streptanthus breweri across this range. Okay. So many others, including Dick O'Donnell, have observed these differences. So here are a few photos showing some of the diversity that you see sort of in nature. So the thing that's tricky about morphological variation is that can be a result of environmental variation. Things may look different just because they occur in different habitats, hotter and drier versus cooler and wetter, or different types of soils. Okay. So what I wanted to do here was use a variety of tools and sort of a variety of criteria for how we might define species to try to better understand potentially hidden diversity or cryptic diversity in this group. So first, typically taxonomists have used morphological definitions to define species. And as I mentioned, particularly in this group, plasticity can be sort of a confounding factor. So in the top left there, this is a picture taken in a wet year in a site that had burned a year or two previously. And you can see those basal leaves of that plant are about the size of my hand. And then a couple years later, not too far away, maybe 20 kilometers away at another site, a very dry year, very harsh serpentine, you can see that entire, well, you can barely see, that entire plant is only about the size of a pen. So tremendous plasticity based on the environment. So I wanted to look at morphology of these geographic clusters, but in a common environment. Second, sort of getting back to the giraffe example and getting to some of my overall interest in the Streptanthus group, is I was interested in biological differences. So that is to say, are individuals from these different geographic clusters interfertile? Can they cross with one another and produce a viable offspring? We also looked at some other important biological differences that determine if these individuals can share genes, flowering time, and mating system differences. I won't get to those today, but we did look at that. And then lastly, we just, again, wanted to use some molecular evidence to infer the evolutionary relationships, but zooming in on the Streptanthus breweri complex. Okay, so what did we find? So here are images of basal leaves that we grew in a common garden. And so what I've done here, I'll back up one second. I went out to about 21 populations across the range, gathered seeds from the field, brought those back to the greenhouse and grew those. So here what the lower leaves look like, and I've arranged them from the top left being the southernmost population to the bottom right being the northernmost population. Next slide. And so what we can see here is a very definitive difference in these leaves. So the southern populations have these rounder, sort of moderately toothed leaves. The north central populations, we see more of a egg-shaped leaf with a very defined toothing. And then as we move to the north, we see more of a heart-shaped leaf with these ears or auricles and almost no toothing. And so very interestingly, given what Bruce told us about Leigh and Justen told us about Caulanthus, is these lower leaves also wither. So these are not really visible on herbarium sheets. These are only visible sort of early in the season. So I found that interesting between these different examples. Okay, so they're morphologically different. And I'm not going to get into the nitty gritty here, but basically I did a lot of artificial crosses. So I crossed individuals from each geographic cluster with individuals from all of the other geographic clusters, sort of in this pairwise fashion. And in every case, we saw reduced interfertility when we crossed individuals outside of a geographic cluster. So the blue individuals were highly interfertile with other blue individuals, but displayed reduced sort of seed production when they were crossed with other individuals outside of the blue area. The same for the north central, shown in the turquoise. And then both of the southern populations seem to be interfertile with one another, but not so much with all of the other geographic clusters. So none of these were complete, but we do know from other work that somewhat distantly related Streptanthus can hybridize, but in every single case, the seed production in these artificial crosses was reduced, was lower when we made crosses that spanned geography. And there are also some flowering time differences and mating system differences that I won't get into. Okay, so then this is just a single gene similar to that gene that was quite informative for the species relationships in Caulanthus and has been shown to be informative in all of the Jewelflowers. And so what we see here, looking at this molecular evidence, trying to infer the evolutionary past of these groups, is that the northern Streptanthus breweri clade shares a common ancestor with all of the other geographic regions, as well as some other species. I don't have my cursor, but that's sort of the dark blue, sort of in the top left of the figure. Again, the north central cluster forms a coherent, a distinct group. And then interestingly, what we've called these southern Streptanthus breweri, they actually appear to be more closely related to other species, Streptanthus hesperides and the Streptanthus morrisonii group, than they are to other things that we've called a breweri. Okay, so we have some phylogenetic evidence that would suggest they're different. And I won't spend a lot of time here, but there was mention earlier about as we're learning about new technologies and new ways to sort of ask if things are clustered or not clustered. Here, instead of just looking at one single gene region, a handful of potential differences, here we've interrogated the entire genome. So we're looking at thousands of different places in the genome, and we actually see even a little bit of a finer differentiation. So the north and the south populations are clustered, as all of the other evidence suggested, but we also see maybe some more differentiation in that north central geographic cluster that we may have missed with other methods. Okay, next slide. So last slide. Basically, I just wanted to say that depending upon how we define our species and what tools we use to look, we often can find differences. So morphology and biological differences that are actually potentially leading to reproductive isolation. All of these things can be sort of subtle, and taking the time to really understand what is what can inform our basic understanding of biology and also our management efforts and conservation efforts. So thank you very much. Appreciate it. Those are slides, so I don't know if we have any more time or not.
We're out of time. I'm going to move on to Shannon's presentation. And it has a little bit of I think we're missing a front slide here, so I'm going to stop sharing and just go straight into.
Can you is my video be able to go on too?
I'm going.
Oh, okay, there we go.
Directly to yours, Shannon, because it is. Hold on just a moment. It played earlier, but I think we've got a little bit more bandwidth or something going. And give us just a moment and we'll change over into you.
Okay. Well, while we wait, Shannon Still, I'm at the UC Davis Arboretum in Public Garden, and I do plant conservation research amongst a lot of other things, I guess. But Schultzia is something I really enjoy. So spent a lot of time on this. A lot more work to be done. It's not playing yet, correct?
It's under Poppy Puzzles.
Yeah. Oh, is it on? Oh,
On just a moment, and I am
okay,
going
yeah.
to share it because it didn't. There we go from beginning. Oh. Let's see. So if there's an animation, it still won't play, I'm afraid.
Okay, yeah. I just have a few little things. Thank you. So today I'm going to be discussing Schultzia and some little issues I faced looking at this. Much like Leigh and Justen and Kyle did all discuss, there's areas where we have trouble identifying some things, and there's a lot of different things we can do in order to solve that. But what I found when I started looking at Schultzia, there's a lot of confusion. There's a lot of things that are misidentified specimens in herbaria. There's a lot of misplaced specimens due to wars or earthquakes and fires, including some of the type specimens. The nomenclature can be very confusing, especially in a group like this where it has almost a couple hundred different species have been described. And that leads to some imprecise descriptive characters, which then feeds back into that misidentified specimens. Go to the next slide. So I guess I guess this Schultzian excessiveness has happened because, well, it started with the original Green New Deal, and that's where E.L. Green, he named 112 species of Schultzia in 1905. 14 more taxa were added a few years later by someone from Germany. And overall, in the past, 180 years since the Schultzia has been described, there's about 191 names and 160 type specimens. So there's been a lot of work. There's probably a lot of things that could still be looked at, or species could be resurrected. And some of it could be valid and some is probably not going to be. But regardless, that what he started in 1905 has led to like a whole century of confusion. Next slide, please. So, but this caused issues. So this is Green, and he was at Cal Academy of Sciences as a botanist, and kind of started the herbarium at Berkeley. And he was the the mentor to Jepson, who I think you guys have heard of. But he was often criticized for naming a lot of new plants, you could say. And if you're talking about 112 species of Schultzia alone in 1905, he really knows how to split things out. But his rebuttal was always, as to the leaves of the Schultzia, the phytoptically blind will say they are all alike. Next slide. And he got very testy, I guess this is maybe a way you would have subtweeted back in the day, where you're trying to tell people something without directly addressing them. But he said there's three types of people: those who can, if you put two species out in front of you, you can tell the difference. And the two of Schultzia. And the other people, if you put them out, if he pointed out the differences, you could tell. And then the third group are the people that just kind of hate you and they're not going to agree with you anyway. So he faced a lot of that, but did all he did. But next slide. One of the people who didn't criticize him too much, at least when he was alive, was his protege, Jepson. And so Green passed away, I think in 1915. He is by then out at the Catholic University in DC. But when Jepson, 98 years ago, wrote A Flora of California, he said, before going further, it should be emphasized that the amount of variation in Schultzia is not by any means as great as would naturally be supposed from all of these specific segregates that people have published. So he's saying there's too much. You guys, like, you're naming things that are probably plastic variation. Like this, you can imagine the Streptanthus that we just showed. It's that type of thing. You go back to the same population, could have something that look completely different. So he grouped at least 90 of those things into Eschscholzia californica alone. There's about 12 taxa when he did this Flora California. Next slide. So, wow, okay. So now, before I started my work, there was 16 different taxa, and now there's a lot of work to be done on these, especially in Californica. So there's, and I know there's some people doing genomic work, whole genome sequencing with the Eschscholzia californica. There's people doing work in looking at common garden, and that stuff's been done in the past. And so if we get more stuff, we may focus on that more. Today, I'll just be talking about Minutaflora and Parisiae, or that group of 3 taxa Minutaflora, and then the Parisiae. Next slide. So why these are interesting, these are all desert taxa, and there was four species in a group, and I ended up naming a couple, and this is the part of the cryptic issue. So we have this hexaploid Minutaflora, very small flowers. Next slide. That's Minutaflora. Then we had Minutaflora cavillii with like a medium-sized flowers. These are tetraploids, so different ploidy, and they're in a smaller geographic range than Minutaflora, which is very widespread. Next slide, please. And then we had another species, one of the 2 named since like 1922 species that have been named is this Twisselmanii subspecies. Twisselmanii found around Red Rock Canyon State Park. And that plant is a diploid, and it seems to be related to Minutaflora. It's got the fairly same characteristics, but the flowers are much larger. It's a diploid. Next slide. But where we get into interesting, well, then every other flower that had of Eschscholzia that was in the desert, in the Mojave or down south, was you go next, go to the next slide, Parisiae. So every flower that had a larger flower was called Eschscholzia Parisiae. And this is when you start to see, well, these ones had a little bit longer buds, a little bit longer bud tip, and that's how they were explained as different from Minutaflora, which had smaller buds, a shorter bud tip in comparison to the whole thing. Now, I probably had these a little bit out of order. My next 2 slides, you can go to the next one. These are the 2 taxa that I named. And so I'll show you the images first, and then I'll kind of go through the morphological characters and how we got to there with the molecular and morphological evidence. So this is Eschscholzia andrewi, and this is I always wonder, do I have cryptic taxa actually? Because this plant, as Bruce mentioned, I think early on, these things have been collected in the past. This was probably, some of these plants were collected probably 80, 90 years ago, or by Green or someone, but they were never named as something different, which doesn't automatically make them something different. But these look sort of like Minutaflora, but have large flowers. So they were typically described as Eschscholzia Parisiae. Next slide. And then there's another plant, the Eschscholzia papistillii here. And this one also had very long buds with like a long bud tip. But the tips are longer than the previous plant that was described, the andrewi. The interesting thing about this is looking at the bud close up, you can see it kind of has a receptacle rim. And if you look down on the plant, it's almost like Eschscholzia californica. And so if you're in the past were to try to key this out, it would key out the californica, because that's the first part of the couplet in the manual. So I have a revised thing, but we don't have that yet. And Bruce will get that into the Jepson online soon. So we have these are the 2 new taxa. And now next slide. How did we get there? So one thing I want to say is I guess what's going to be the take home of my presentation is we have cryptic taxa, but a lot of times it's everything that has gone on before us has kind of biased us toward how we describe a plant. So everything I've learned and I started to look at like those when Eschscholzia was first described in like 1826, all the characters and the things how they describe these desert taxa, and they use the exact same characters every time. They talk about bud length and how rounded the tips of the little leaf segments are, or if they're pointy, how many leaf segments are on there, or there's not that, or how many. They've had a hard time kind of choosing good characters. So when I started to look through, I was very confident. I'm first going to the herbaria to look at plants. I'm like, oh, I kind of got this. I'm looking at the previous descriptions and looking at the keys, and I can really key these things out. I feel confident. And then you talk to someone who's an expert in the field. Maybe you talk to Jim Andre, and he says, I have trouble with these, and I'm kind of curious, like, well, he's an expert. Why would I be no better than him? And it was like, but I really think I do. But once you go out in the field, see the plants, you're like, oh, I am having trouble in certain areas, just like looking at these plants and putting them into a bucket somewhere. And eventually, after kind of looking at herbarium specimens in the field, and I first started with a lot of herbarium specimens for my phylogenies. And there's one animation here, so if you can kick to that. What I noticed here is that I think I had paraphyly, because, and I think there's one more, kick to that. So what I was seeing is I had a Schultzia minutaflora and Parisiae, ones that look sort of like either of those, but they're closer in the tree to the Parisiae. And I had some Parisiae that were ending up down as things labeled as Parisiae that were going down to the minutaflora clade. Next slide, please. And so that's a kind of a sign of trouble here. We had got really bad. I was even cloning plants, the things where I take something out of a packet on a sheet, and ultimately we figured out, okay, there's actually different plants on that sheet. And so when I pulled something out of the packet and I had to clone it, and they would come out in different parts of the phylogenetic tree. You might have minutaflora close to the Parisiae, and then other ones close to the minutafloras. And so click to the animation here. So all this is showing is that, okay, these are probably all Parisiae that got, and I go back and look at these plants and kick to the next animation. I look at these and I say, oh, these do look more like a Parisiae, and the ones in the gray box that label Parisiae do actually look more like a minutaflora, but they do have very large flowers. Go next slide. I think you might, yeah, go through this and then go to the next slide. Next slide, we'll just go through this one. So we eventually realized, okay, we have something new. And this wasn't just like I ran an analysis phylogenetically and I thought, oh, okay, I have these and I name them. This was a lot of going back and forth. And this is the refined view of what I've seen. But all these things labeled EZ were this new taxon Papastillii, which looks sort of like, or very similar to Parisiae. Then you got this Andrewiae is very similar to all the minutafloras except a larger flower. And it's easy to show you here, like in an illustration, oh yeah, those kind of look different. If you like left and right, like Andrewiae versus Papastillii, all the characters, okay, that's easy. But these fit in with all the other taxa that were already described. So how do we tell them apart? Go to the next slide, please.
1 minute.
1 minute, okay. So now why I stressed like what the things we looked at in the past. So we had basal leaves that we really focused on. Are they the segment tips? Are they point acute or obtuse, or how many are there? They're fairly constant across how many segments there are, but this can be a problem, just like in the Streptanthus, where some things in a good year are very big, the leaves are large, they have more segments, but they've always got more pointy tips, as you see on the right side of these figures. The right two are always the kind of the Parisiae pair of plants. Next slide, please. But then one thing that was interesting that I'd never seen use it as descriptor is actually the Cauline leaves. And this is why I say like, it's important to know what your biases are, because this is the thing that actually really helped me to distinguish when I see a plant and I kind of get this gestalt in my head. It's mostly because I see these Cauline leaves and what we're seeing here, where the Cauline leaves, the segment number, go to the animation here. The segment number on a minutaflora, which is the top right illustration here, has a lot more leaf segments, 3 or 4 times as many as the Parisiae or Papastillii for the same length of thing. Go to the next slide. The bud lengths are pretty typical, like we'd see Parisiae, Papastillii larger, but then I have to distinguish between those Papastillii and Parisiae then. So next slide. So then we could go to a new, we hadn't really looked at, is the receptacle. This hypanthium isn't large, but I talked earlier, the rim is there. on most of these Parisiae. So if you so that means the key needs to be redone so we don't start putting these things all into Californica, but that's a good way to help distinguish between the Papistillii and Parisiae. And there are then the other characters that we've gone through are going to distinguish between them. Next slide. So I won't spend a lot of time here, and I can share stuff with people, but the Schultzia, we've gone to like now 18 taxa, and go to the next thing. I hope, next slide. I hope that the three important people like in this stuff, the Green and Kate Brandigy and Jepson would be happy, like they might actually now be able to see this and see that. So and take home messages like with taxonomy, the past work is going to influence our future perception of what things are. So you really got to go back, and sometimes it's rebuilding all those things you're going to look at for like those morphological characters and trying to find something new. Right, thank you.